Okay, so in this PowerPoint, we're going to go through the basic cell structure. So looking at all of the organelles and how they work. This is the first part of Unit 2. All right, so tour of the cell. This slide just kind of goes through the things that you need to know. So the idea here is that you understand the structure and function of the subcellular components, basically the organelles, how they contribute each to the overall function of the full cell. We're going to talk about how that allows them to capture, store, and use energy. At a certain point throughout the unit, we're going to talk about surface area to volume and why there's limits on cell size. And then specialized structures that basically help exchange molecules with the environment, whether it's taking things in, giving things off, communicating, and whatever. Okay, so there's two types of cells, and this is what a lot of you were asking about Unit 1 because you saw these words in the FRQ examples. Prokaryotes. Pro means before, and karyo means nucleus. So a prokaryote is essentially bacteria and archaea. It's two different categories. Whereas eukaryotes means true nucleus. These are the ones that have nuclei in the cell itself. And hopefully you guys remember from bio, like the nucleus is the center or the brain of the cell, so to speak. But many things fall into that eukaryotic category, like protists, fungi, plants, and animals. And you can see on the left is an example of a prokaryote. Oftentimes they have their nucleic information just kind of free floating around, whereas on the right there's a defined nucleus. And in this case, there's even a kind of nucleus of the nucleus called a nucleolus. All right, so here's the prokaryotic cell in detail. You can see that they have what's called like a nucleoid. It's not in a nucleus, it's just all of the spaghetti-like genetic material in this case is lumped together. They have other organelles too that we'll end up talking about. So they have ribosomes. So that's a commonality between prokaryotes and eukaryotes. They have plasma membranes. They also have cell walls, and they have kind of an outer layer called a capsule, which is kind of like an extra layer of protection around the end. And they have other extension things like cilia and flagella that we will end up talking about at a later point. Okay, so I'm just going to click through this and then talk about it. So as I mentioned, prokaryote means before kernel or before a nucleus. They don't have one. DNA is in that nucleoid. They have a cytosol or cytoplasm, just like eukaryotic cells. They don't really have any other defined organelles inside of them besides ribosomes. They are usually small in size. They are considered primitive. Having a nucleus is considered more advanced. And that is the bacteria and archaea, which are like old bacteria. Whereas eukaryotes, they do have a nucleus. They have a nuclear envelope, which provides kind of an extra level of protection and transport. They have a cytosol, just like prokaryotes. They have other membrane-bound organelles. So ribosomes are not membrane-bound organelles, but eukaryotes have other things that we'll end up talking about. Eukaryotes can be much larger in size and much more complex, like a plant and an animal cell. So the first is the nucleus, and its job is to control the cell, kind of. Like I said, it's the brain of the cell. It has the DNA, and oftentimes, as we will learn later when we talk about how proteins are made, it is where mRNA is made as well. It has a double membrane, so it kind of has its own cell membrane, except it's called the nuclear envelope. It's the membrane that surrounds the entirety of the nucleus. That membrane is continuous with the rough endoplasmic reticulum, or ER, which I will explain what it does later. It is permeable in the sense that it has these little tiny holes called pores, and it can then control what enters and leaves. So it's not like anything and everything can go in and out of the nucleus. Chromatin is kind of that spaghetti-like stuff that you saw in the bacterial cells. It's got DNA. There's also proteins mixed in, and chromatin is kind of the unpacked form of what will become chromosomes. It's the uncondensed version. The nucleolus is where the RNA subunits live. So we're going to learn about different kinds of RNA. There's mRNA, tRNA, and rRNA. rRNA is responsible for making up the ribosomes. So those subunits are formed inside the nucleolus, and then they can kind of go wherever they need to go from there, but that's where they're made. Here's an example of what a nucleus looks like on a electron microscope in terms of the different membranes and the envelope and the pores and things like that. Here's kind of a more zoomed in picture of what those little pores look like. So I kind of think of it as like a wiffle ball. It's a central point that has holes around the outside. Here's just another picture of kind of what that looks like. You can see kind of the chromatin in the middle. The nucleolus is in the center where the ribosomal subunits are made. You can see that chromatin kind of looks like this. It is DNA kind of wrapped around proteins that we will learn about, but it looks a little bit like balls on a string. And then, as mentioned, the nuclear envelope here that contains a double membrane is continuous with the rough ER. So, ribosomes. What is their job? Ribosomes are the site of protein synthesis. They are in all life forms because if you can't make proteins, then you can't be alive. They are composed of both rRNA, which, as we mentioned, those subunits are made in the nucleolus, and some protein. And it's composed of two subunits. There's a big fat one and a small one. And we learn a lot more about this when we talk about protein formation. But they can either be free, meaning they're just floating around in the cytosol or the cytoplasm, and they are still protein factories inside the cell. So when, information comes to them, they are able to produce proteins. There are also bound ribosomes, and those are the ones that are embedded in the endoplasmic reticulum. And that's why when we get to it, there's going to be a rough ER and a smooth ER. The ones that have bound ribosomes are rough ER. But in the grand scheme of things, the endoplasmic reticulum is kind of like a packaging system. So for things that are going to be exported from the cell, the genetic material can kind of leave the nucleus, go through the ER, and as it is, proteins are being made, then it will eventually be packaged up and exported. Here's an example of what those ribosomes look like. You can see that there are free ones kind of dotted all about. You can also see the ER, kind of those ribosomes embedded in to the ER. And you can see just a graphic image of kind of what a ribosome looks like with the big unit and the small subunit. So this basically gets us to the transport system, the endomembrane system. And you can kind of see from the picture, the rough ER that we were just talking about, it is attached to the nucleus, and it has rough studs on it called the ribosomes. And then the area that doesn't have those is considered the smooth ER. And that eventually there's going to be other transport involved to export those proteins that we need. But it's the trafficker, protein traffic. It also does some metabolic functions that we will learn about. So there's lots of sacs, and it's kind of like this never-ending conveyor belt. There's the rough ER that have the ribosomes. Their job is to package those proteins and send them off to the Golgi apparatus, which we will learn about, and make replacement membrane. So they, help to replace and replenish the membrane as well as export things. The smooth ER is smooth because it does not have any ribosomes on it. And it is the part that makes lipids. So as we learned in the first unit, those include the steroids, the triglycerides, other phospholipids. They are able to break down carbs. They also act as kind of the detox agent. So certain things of drugs and poisons can kind of be broken down there, as well as the smooth ER is the source of calcium, which is going to be important in certain cell types later on that we talk about. The endoplasmic reticulum here, again, you can kind of see what that looks like. The smooth doesn't have the ribosomes, the rough does. They kick off these little tiny guys that we'll eventually talk about called transport vesicles. So from the ER, things get packaged up and kind of butted off in little packages called vesicles and carried to other areas. And you can see here in the picture the smooth versus the rough based on the presence of ribosomes. All right, the Golgi. Its job is to basically fold and modify in any way the new proteins that are made. So in the conveyor belt of things, the genetic material is made in the nucleus. The ribosomes on the rough ER are able to turn that genetic material into proteins as it travels through, and then it goes to the Golgi. So the Golgi is really responsible for ensuring that secondary and tertiary and quaternary structure and any modifications that need to be made are done in the Golgi. And then it kind of packages them up and sends them where they need to be. There are two sides of this smooth ER. The cis face is the receiving side. So that's where it receives the little vesicles that have been packaged up from the ER, and then it comes in and it works its way through, and then it'll go out the trans face. So there's kind of two sides. Cis means same, so it's on the same side as the endoplasmic reticulum, and then trans meaning the opposite side, it's sending it away to wherever it needs to go. Another type of organelle that's important is the lysosome, and we learned about like hydrolysis means to break with water. Lys means to break, so it makes sense that a lysosome is responsible for breaking things down. So it's kind of the cell digestion or garbage disposal. It executes apoptosis when certain cells need to die, as well as recycling material as needed. It contains hydrolytic enzymes, so that's where hydrolysis comes from. It's the enzymes that make that magic happen to break things down. You can see here just kind of what that looks like. So at the bottom, on the left, we have kind of cell eating. So phagocytosis means cell eating. So in the bottom left picture, food for whatever reason is being brought into the cell in a vacuole. Again, a vacuole is just a fancy way of saying like a little package of things. So if we want to actually incorporate this, then the lysosome comes in and brings over its little digestive enzymes, and now we can actually break down whatever it is. We also have autophagy, where we have a vesicle that might contain certain material or organelles that we want to get rid of or whatever it might be. The lysosome can then take care of that and transport its digestive enzymes and break that down. Vacuoles. Vacuoles are just bags of stuff. So their job is to store. They're like the Ziploc bags of the cell. They can store food, water, minerals, poisons if necessary to try to break down later. They have all kinds of stuff. They are membrane bound. They are found a lot in animal cells for food vacuoles. There's a giant one in water, there's a giant one of water in plant cells. So it's most readily apparent there. You can kind of see in this particular picture, it's huge. The animal ones and the ones that store food and things like that are much, much smaller. But for plant purposes, they need to have a lot of water. So that's kind of what that looks like. All right, so transport. From the nucleus to the ER, the rough ER, and then it can get packaged up sometimes to the smooth ER depending on what it needs to do. Then it goes into a vacuole over to the cis Golgi, where now that protein that's just been made can get packaged up and folded and get ready for transport, where it can now go out the trans Golgi. Oftentimes it'll then fuse and go off into the outside of the cell through the plasma membrane, but it could also go to other parts of the cell too. And that's where here it's fusing with the plasma membrane if it's going to now go out into the environment. The next organelle is the mitochondria, and you've probably learned, or it's ingrained in your blood, that mitochondria is the powerhouse of the cell. And it's called that because it is kind of like the battery. It's where cellular respiration takes place, and cellular respiration is where the body is breaking down like the carbohydrates that we eat to provide us with and make ATP, our energy source. So that's its job, to convert the food that we eat into our energy source. It's got a double membrane system, as you can kind of see in the picture below. It's got an outer membrane and an inner membrane. The folding part is called cristae, and that's the folds of the inner membrane. There are enzymes that like to live in there that do ATP production. And the way that it folds back and forth is really a way to increase surface area, and that increase in surface area allows more ATP to be made. So it's just kind of an efficiency thing. The matrix is the fluid-filled inner part. So there are kind of like ribosomes just floating around in that fluid that's in the middle. And certain parts of cellular respiration take place in the membrane, and certain parts are in the matrix. A specific organelle that's only found in plants, so that's kind of a key giveaway. This one is only in plants, is chloroplasts. And chloro means green, or chlorophylls are color. So their job is to have and be the site of photosynthesis. And photosynthesis only takes place in plant cells. So within the chloroplast itself, it's got a double membrane. It's got little stacks inside. Each individual, I think of like a roll of quarters, and each individual quarter would be a thylakoid disc. And when you stack them all together, it's called grana. So an entire stack kind of in here, roll of quarter style, is a grana. And then the fluid floating around in there is called stroma. But the key part here is inside those chloroplasts live the chlorophyll. And chlorophyll is one example, but there's yellow pigments called xanthophylls. There's orange pigments called carotenoids. It's the site, we say chlorophyll just because most plants are green, but that's where the pigments live. And the pigments are responsible for capturing sunlight. And that's what fuels that whole process of photosynthesis. And if you've ever not put a plant in the sun, that is why it dies, because it needs the light in order to fuel the whole photosynthesis reaction. So chloroplasts are known as the site of photosynthesis because it's where the chlorophylls live to absorb that sunlight energy to drive the reaction. All right. Now, part of what in the grand scheme we've thought through history to go from getting kind of more advanced cells is called the endosymbiotic theory. And symbiosis is really kind of like a harmonious relationship between two things. And it's once thought that once upon a time, we had an ancestor eukaryote. So it had a nucleus, but not much else. And then once upon a time, that cell essentially ate a mitochondria. It engulfed it. And that mitochondria used to be its own freestanding prokaryote. But now it got kind of absorbed into the cell, and now it acts as what we currently have as a mitochondria. And it's thought that because the mitochondria has a double membrane. So because it kind of maybe had one originally, and then when we engulf, it's got two membranes, which is kind of different than the other organelles. But it can't harness sun energy. So at least one cell at that point then also ate a chloroplast, which similarly was its own prokaryote that could photosynthesize or take energy from the sun to fuel photosynthesis to make food for itself. And then cells were able to then take that in and turn it into a organelle. So that would have taken place in what we now have as plant cells. Animal cells, again, do not have chloroplast. But the chloroplast is another double membrane organelle. So that's kind of why we think that that happened. And it's referred to, again, as the endosymbiotic theory. As I just going through, I talked about it. Another key piece is that mitochondria and chloroplasts have their own ribosomes within them, and they have their own DNA within them. So, for example, parents or moms pass on mitochondrial DNA to their children, which is different than kind of your normal 46 chromosomes that you get when you combine an egg and a sperm together to make a child. And also, fun fact, they reproduce by themselves within the cell. So they don't need the whole cell to go through the cellular division process. they can just do it by themselves. So you can end up having a lot of mitochondria and ramping up chloroplasts also. All of that gives evidence for them kind of once upon a time being their own organisms. Okay, cruising along, we now have peroxisomes. Their job is, I always think of like peroxide because that's kind of a similar chemical that's happening here because it involves production of peroxide, H2O2. It breaks down fatty acids. It can also be the site where alcohol is kind of detoxed in your system. And you can see here, we have chloroplasts, and you can see the grana and thylakoid discs and so on. But then here is the mitochondria. You can kind of see the zigzaggy inner matrix and membrane. And then here's the peroxisome, and it's not quite like a lysosome because it doesn't break down everything, but it uses peroxide to break down those fatty acids and alcohols. Okay, so this is an example of altogether an animal cell. And you can see all the hallmark things. Save the nucleus. It's got the nucleolus in the middle. It's got the nuclear envelope that has the pores in it. That is consistent with the rough ER because it has ribosomes on it. So that's where the genetic material can be made into proteins. Some areas and functions go over here to the smooth ER, where it can make things like lipids. Otherwise, those proteins get packaged up onto the cis side of the Golgi, where they get folded up and modified in any way they need. They pop out the trans Golgi and then might fuse with the plasma membrane or go somewhere else. We also have the peroxisome that we just talked about that has peroxide inside to break down lipids. We have the lysosome, which is kind of the garbage can of the cell that breaks down most things with its hydrolytic enzymes inside. Because this is an animal cell, we have a mitochondria, which is the powerhouse. That's where cellular respiration takes place. You will notice that in an animal cell, there is no chloroplast. There are no central vacuoles. There might be smaller, little lesser vacuoles throughout, but there's no giant central vacuole. There's no cell wall. And not really important right now, but there's no plasmodesmata, which I'll talk about at a later time point. Don't worry about that vocab word. And this contrasts with a plant cell. So many of the things are exactly the same. There's still a nucleus with all of its bits. There's still a rough ER. There's still a smooth ER. There's still ribosomes, still a Golgi apparatus. Differences, there's still a mitochondria. There's still peroxisomes. There's still a plasma membrane, but things that are different. We now have chloroplasts. So you can see those thylakoids and grana inside. Additionally, we have a cell wall that's not present in animal cells. We have that large central vacuole in the middle. And here's what they call plasmodesmata. It's basically like tiny holes in between cell walls so the cells can communicate. Again, you don't really need to know that. But what you're missing here is there's no lysosomes in plant cells. The other things we don't need to know totally for now. It will come into play when we talk about cellular division, but there's no lysosomes. Okay, so continuing on with this idea, it's kind of an offshoot, but cell size and scale. This picture just kind of gives you an idea of the scale of things. So 1 meter is kind of like a little more than 3 feet, I would say. And human height is somewhere in that range. And then you get down to a millimeter, which you know is kind of small, but still seeable on a ruler. And you can see all the way down to about the middle of the micro scale with your unaided eye. And then you start to need a light microscope and then an electron microscope. So crazy small. Micro means times 10 to the 6th. Nano means times 10 to the -9th. So those are some small things, but it gives you an idea of generally the small scale. Most plants and animal cells are somewhere around here at between 10 and 100 micrometers. And then the nucleus is obviously smaller than that. Proteins themselves are orders of magnitude smaller. So the idea here is that cells have to be small. because they have to be able to transport things from one place to another. They need to maintain a large surface area to volume ratio. So we're going to do some activities with this, but keeping in mind that if you have one just super giant cell, it's going to take a lot of energy to move proteins from kind of one side of that to another. And there's not a ton of surface area or contact with the outside environment. So similar to why the mitochondria has all those zigzags of membrane on the inside, it's to increase that surface area for a given volume so that more chemical exchange can take place. And this picture just kind of shows you that if you have one cube, what its surface area to volume ratio might be. If you have one really large cube, how low that is. You can see compared to the single cube, it's really low. But if you have a whole bunch of little cubes that have, independent surfaces, then it can take into account the total surface area to volume ratio. So why do we have these things? Membranes and membrane-bound organelles are able to self-contain metabolic processes and reactions. They can kind of house their own enzymes and make certain reactions take place without it kind of floating everywhere. So that's one of the advantages of having membranes and having membrane-bound organelles, is it can contain certain enzymes that need to perform certain functions. Internal membranes minimize competing interactions. Like I said, it kind of sections them off and compartmentalizes them so they're not all over the place in various forms. The membranes also increase surface area. So if you're trying to make a whole ton of proteins, that's why the rough ER in this picture means that you can have more enzyme, and more enzyme means more reaction. So just more product. So the idea is you're maximizing your efficiency. You're making as much of something as you can in the limited volume that you have. So kind of here's an animal cell example. This is what our intestines look like. We have a ton of kind of folds. They're called villi that are also popping out of the intestinal wall. So between the kind of ribbing back and forth and all of the tiny little villi, it's basically just maximizing all the amount of surface area so that as our food and our digestive material works its way through there, we are absorbing as much possible stuff as we can. Plants, one example is root hairs. So if you look really, really closely, a lot of plant roots, they have tiny little, looks like fuzz hanging off of them. But the idea is those tiny little root hairs help to increase the absorbing surface area of water. So they can suck up more water. Even though they don't have really long and strong roots, they can absorb more water and minerals to kind of sustain the plant. All right, so we're to the end of this PowerPoint. At the end, there's kind of a review of just what the structure and the function is. So nucleus is kind of the brain. That's where in the nucleolus, the ribosomal subunits are made, and there are pores to regulate entry and exit. Ribosomes, they can be either bound or free, depending on where they are in the cell, and they are the site of protein synthesis. The ER is the membrane-bound system that's job is to make the proteins, whereas at least the rough ER. The smooth ER has a variety of functions like making lipids, breaking down carbs, storing calcium, and detox. The Golgi is the packaging facility of the cell. It helps to fold up the proteins and package them wherever they may need to be. The lysosome is the garbage truck of the cell that has the hydrolytic enzymes to break things down. Note that those are only in animal cells. A vacuole, which is a membrane-bound vesicle. Those are small and tiny in animal cells, but there's that one giant central water-containing vacuole in plants. The mitochondria is a double membrane-bound structure that is responsible for cellular respiration, which is where we turn our food that we eat into ATP energy. Another double membrane organelle is the chloroplast. That's the site of photosynthesis. Again, the pigments like chlorophyll that are inside harness the energy from the sun so we can do that. The peroxisome contains peroxide, and its job is to act as another metabolic component, but it only has a singular membrane. So that's it. That's the end of this presentation.